Can new research improve Ewing sarcoma treatments? The question of whether new research can improve treatments for Ewing sarcoma is an important one. Ewing sarcoma is a rare and aggressive type of bone and soft tissue cancer that primarily affects children and adolescents. Thankfully, ongoing research is making strides in advancing treatment options. These developments aim to improve survival rates, particularly for patients with advanced disease, while also reducing long-term side effects from current therapies. One exciting area of progress is targeted molecular therapies. Researchers are working on novel molecules that specifically degrade proteins essential for Ewing sarcoma cell growth. For instance, a small molecule known as DGGA3 targets a transcription factor called ETV6, which is crucial for tumor survival. This molecule belongs to a class called transcription factor proteolysis targeting chimeras. These targeted approaches could offer new treatment options for cases that do not respond well to standard therapies like chemotherapy and radiation. Immunotherapy is another innovative field making waves in Ewing sarcoma treatment. Advances include developing chimeric antigen receptor T-cell therapies that target proteins highly expressed on Ewing sarcoma cells, such as STEAP1. Since STEAP1 is found in over 90% of Ewing sarcoma tumors and is minimally present in normal tissues, CAR T cells engineered to attack STEAP1-positive cells have shown promising results in early studies. This method aims to harness the immune system to specifically attack cancer cells, providing a more tailored treatment option. Research has also uncovered how the EUS-FLI fusion protein, which is characteristic of Ewing sarcoma, disrupts the cellular skeleton and promotes tumor spread. Targeted treatments that interfere with this process could help prevent metastasis, a significant factor in poor prognosis. Additionally, Ewing sarcoma cells relying on DNA repair mechanisms to survive damage from chemotherapy and radiation. By inhibiting proteins involved in DNA damage response, such as poly-ADP ribose, polymerase, researchers are exploring ways to make cancer cells more vulnerable to these treatments. Combining PARP inhibitors with radiotherapy is currently under investigation to enhance treatment effectiveness. Clinical trials are also testing combinations of existing chemotherapy with new drugs like regorafenib, a tyrosine kinase inhibitor. Early-phase studies indicate that combining regorafenib with standard chemotherapy is feasible and well-tolerated in young patients with multimetastatic Ewing sarcoma. This combination is moving toward larger trials to evaluate whether it improves outcomes compared to chemotherapy alone. Ongoing randomized clinical trials continue to explore adding new agents to standard chemotherapy regimens to improve survival in newly diagnosed patients without metastases. These trials are essential for establishing the safety and efficacy of new combinations before they can become standard care. In summary, ongoing research is broadening the treatment landscape for Ewing sarcoma through targeted molecular therapies, immunotherapy, disruption of tumor spread mechanisms, DNA repair inhibition, and novel drug chemotherapy combinations. While standard treatment remains surgery, chemotherapy, and radiation, these innovative approaches hold promise for improving survival rates and quality of life for patients facing this challenging bone cancer. It is crucial for patients to seek care from specialized medical professionals and consider participation in clinical trials when appropriate.